2015, Chinese researchers removed part of the DNA of an human embryo, which is related with thalassemia beta, a disease that causes serious anemia and that can be fatal. That genetic manipulation is called CRISPR. It was very polemic and opened a lot of ethical discussions. Beyond it presented several changes other than those targeted by scientists. However, it is a consensus among scientists that in a short time, the technique may be safe enough to be widely used in human embryos. These changes can modify the next generations, intervening directly in the future of our species. Some scientists believe that the practice would be used, in addition to avoid diseases, to manipulate physical characteristics such as eye color, hair type, or even skin color.
Transgenic, the insertion of one or more genes into the genome of the organism, being two expressed genes not sexually compatible. Cisgenic, it is a term used when an organism is modified genome through this insert of genes of species that can be crossed naturally. Genetic silence or RNA interference, a cellular mechanism that inhibits the process of protein production by means of the shutdown of the genes that express it. Cloning, it is a production of genetically identical copies of a gene, a cell, a tissue, or even an individual. And CRISPR-Cas9, one of the newest genetic engineering techniques being applied. CRISPR-Cas9 edits certain DNA sequence of an organism, removing one piece and replacing it with another. Okay.
The genetic engineer laws varies widely by country, and the most laws refer to food because the biggest area in the genetic modification currently. Institutions that conduct certain types of science research must obtain permission from government, authorities, and ethical committees before they conduct any experiments. University and research institutes generally have a special committee that is responsible for approving any experiment that involves genetic engineering. Brazil is the second largest producer of genetic modified food, and the National Biosafety Technical Commission is responsible for assessing environmental and food safety and prepares guidelines for transport, importation, and field experiments involving genetic modified products.
Another area of genetic engineering is the modification of animal DNA. Genetically engineered animals are approved for 6 categories. Used for biomedical research, smaller mammals can be used as model in scientific research to represent other mammals. Used to develop innovative kinds of fish for environmental monitoring, and used to produce proteins that humans lack. This can be for therapeutic use, for example, treatment of disease in other mammals, used for investigating and finding cures for disease, can be used for introducing disease resistance in GM breeds, and used to create manufacturing products for industry use, and used for improving food quality. For humans, genetic engineering is still a big taboo, and it doesn't have many laws because of this discussion.
Hello everyone, we're here with Thiago Matos, and he's postdoctoral at Unicamp, and he's working with IRX3, that is a gene. So he's talking with us about engineering manipulation. So Thiago, techniques of manipulation of genes were know that CRISPR it's the most used. Why this technique specifically is the most used?
This technique is really new, yes, but some techniques before come to get some improvements in the CRISPR. For example, Talent. Talent was before the improvement of CRISPR-Cas9. Talent was really used, but CRISPR-Cas9 comes with a new generation of knockout or not in the industry, in the science, because it could be faster and cheaper.
These experiments with genetic manipulation generally was made in animals. In 2018, a Chinese published an article that he was studying this manipulation in humans. What changed for he becomes student this?
The scientist world is not really approved in total this kind of approach, because it's really new technique. We don't know the problems that kind of manipulation could be in the future, and it's not really clean, the studies, and we don't know exactly what this kind of things come in the future, or if it's really true for us.
You start to talk about our next question because in 2018, Alan Bradley published an article, and in this article he said that this manipulation was causing trouble and mutations unexpected.
Yes.
So, do you think it's worth to use this manipulation to treat disease?
It could be used maybe in the future, but today it's really new. We need to be very careful about it and working a lot to get some information, something more strong to improve or to change this technique maybe, because science is this, we need to do some manipulation, some modification of the technique to improve better and better technique.
So, Thiago, what the biomedical science can do?
What biomedical science can do? Basically, we are studying in the graduation to be a doctor in the laboratory, working in the diagnostic, helping the analysis about blood and other fluids that could improve or get some directions to know if the patient is better with treatment, or if some disease could be work for good to the patient.
Okay, and what the ethics code talk about the manipulation of genetic?
Yes, it's really difficult because it's a word knowledge that we need to be really careful about some kind of manipulations. We need to start to do this manipulation in bacteria, in cells, and after it's in animals, and to prove that improvement and that manipulation in the gene, this kind of manipulation could improve or could be better for the health. We need to be really careful about this kind of approach.